New, she has an area of interest: neurodegenerative diseases, Huntington diseases, Parkinson disease, and amyotrophic lateral sclerosis. She has a visiting scientist in the University of California, USA. She has achieved a position of the project scientist, research specialist, postdoctoral fellow at the Department of Development and Cell Biology, University of California, USA. She is a member of Genetic Society of America, USA. She has published several research papers in peer-reviewed journals like Science, PNAS, Development, JCP, etc. Presented our work previously and recently in the several international conferences. Now we have our one invited speaker, Professor Rajnikant Mishra. He is a professor at the Department of Zoology, BHU, Banaras. His area of interest is understanding the biology of diseases associated with the CNS, eye, spleen, and pancreases and lymph nodes. Contributed to the understand mechanism of the functions of paired box family, transcriptional factors in the brain immunity, adult neurogenesis, neurodegeneration, diabetic neuropathy, Parkinson's disease, ocular defects, and lymphoma are the well recognized. Sir, we welcome you here for this session.
साउंड नहीं आ रहा है, इसको डिस्प्ले में लगाइए. नहीं, इसको डिस्प्ले में. हाँ. वहां कुछ होगा, पता नहीं चला. आवाज नहीं आ रही. बुलाइए वो साउंड वाले. Anyway, I think I shall be audible. So, thank you very much for your kind introduction, and I am thankful to organizers, specifically Dr. Anju and Dr. Alok, for giving me opportunity to share some of our findings and the area which we work on. And a topic which I would like to share with you is related to the PAX genes and some of its impact related to the neurological disorders. So I would like to take about 15 minutes which is given to me, and first 5 minutes I would like to introduce you about what is PAX and specifically one of the PAX gene family and some of its detail. Next part I would like to work on that is on the microglia, some of its observation on microglia, that how it is functioning and what is our observation. And last part I would like to explain something about what is our observation related to the interactome, that how these interacting proteins they are functioning and how they are helpful to make us understand about this biology. And in last part, since the theme of the conference is the nutraceuticals, I would like to share some of our recent hypotheses and some of our work which is related to the role of these nutraceuticals, which are specifically related to the neural regeneration or the microglia biology. So, to introduce you to some of you who are unaware about the PAX genes, in vertebrates there are 9 PAX genes and which are called as PAX gene family. And out of these 9, today I would like to share some of our observations related to the PAX6. Specifically, PAX6 is expressed in most of the part of the brain, Today, I would focus mainly on brain. Other than that, it is well known for the eye development, and rather in the beginning it was identified as an iridia gene. And before that, it was known to be causing a Wager syndrome, and which was related to Wilms tumor, aniridia, and urogenital diseases. So out of that, in 1991, when it was cloned from human, then I was fortunate to work with that group. And then we have some observations related to that. So other than its development effect, what is our interest is like how it is and what it is doing after the development. So very first observation is that it is required for differentiation of the So it is required for the differentiation for neuronal cells from the neuronal stem cells. Other than that, it is also required for the differentiation of different cell types. So at a critical point when there is no PAC6, there is no differentiation of neuronal cells and also no differentiation of other neuroglial cells. Other than that, it is also required for the neuronal migration. So those mice which are not having this PAX or they have low level of PAX, they have defect related to neuronal migrations. Other than that, it has different other properties, including almost all part of the brain, it is expressed. When it is like if you go through the mutation database, which is one of our area because we work on the functional annotations, and in that one, what is our observation and through the literature that if there is heterozygous mutation, there is no eye, no brain, no any other specifically sense organs. Other than that, like the patients which are having the mutation, they have a glucose intolerance. When there is homozygous mutation, severe brain abnormalities are there, including microencephaly. And if it is haplosufficiency, several brain defects have been observed, like loss of anterior commissure and loss of olfactory lobe. Other than that, like some of the observations like 3' UTR mutations, those patients, they are having autism. So this way, it is affecting almost all functions which are very critical for the survival or normal status of any of the organism. So what is observation is like if there are mutations, it is related to eye, brain, pancreas. Today I would like to mainly focus on the brain and associated disorders. What I try to tell you that if there is knockout, in the knockout mice, they are not having brain, no eyes, and loss of specifically alpha cells. And other than that, mostly they are embryonic lethal. So what is other observation that looking into the database, that other than those known direct effect of the gene, there are non-aniridia phenotypes. So this was our interest that yes, what are those and why they are happening and why this regulator protein is affecting several other organs beside their gene which were classically known. So we focused on that and we try to look into the interacting proteins through the database search, including the string. And what we could find, like it is having putative interactions with most of those proteins which are required for the to maintaining the functional anatomy of brain. And many of them we try to characterize through different aspects like chip, and co-IP, and including some of them by creating site-directed mutagenesis and looking into their functional status. So how they are interacting in most of the models. Then based on like a cytotrap is2 hybrid system, which is a little bit modified classical 2 hybrid system, of almost all those work that it is interacting with most of the critical enzymes, including signaling components. So how a transcriptional regulator, which is supposed to be in the nucleus, how it is functioning as a signal transduction molecule, or how it is affecting the signaling. So after looking into the detail of this protein, it has two DNA binding domains, pair domain and homeodomain DNA binding motifs. Other than that, its transactivation domain is rich in proline, serine, and threonine. So all of these, they are having phosphorylation sites. So these phosphorylation status happens to be one of them, which is critically affecting those signaling, IG signaling molecules. So it was very hard to establish like how a transcriptional regulator, which is present in the nucleus, how it is functioning to be there in other way. Other part which I try to explain you here is that microglia. So the neurons glia, they are expressed, and then our interest was on microglia. One of your colleague who is here, he started this work in our laboratory, microglia related, Dr. Susan, and he tried to take it as a challenge because many a time we were getting an unusual observation. So after several such unusual observations, which we thought that it is an artifact, we could identify that it is the different forms of the microglia. So that way we started working on it, because it has a multiple role depending upon the microenvironment. Microglia, as many of you are aware, they have a different properties, right from its daily maintenance to activation, and also the activity of the neurons, regulation of those neurons based on different type of inserts and the amount of inserts. So like we try to observe some of it, and I would like to share a few of those observations today with you. So this is an overall hypothesis and a working hypothesis, like PAX, which is required for neurogenesis, also for the neurodegeneration, specifically PAX6. It is expressed in neurons and glia contains both microglia and astrocytes. So how this environment, they are very critical, whether PAX6 itself is regulating or the environment of PAX6 depends upon like how and which gene will be regulating each other. So we try to work on it, and with a hypothesis that yes, brain being an immunologically privileged organ, whether it is involved in the brain immunity and homeostasis. So we had very good observations related to it, and we will I will try to share some of it. But we could find another like microglia, which is identified. A marker of microglia is one of a potent marker EBA1, and other than that, like a mature microglia is identified by a marker MM119. So we have worked on both, and what we could find, like both PAX6 and EBA1, they are involved in somehow a common type of activities. So that was our another interest, and we try to explore a little bit more related to that one. So we could publish this like yes, PAX6 and EBA1 both interact, both as a protein, and also they interact through their promoters. So we try to work on it, And this was our landmark observations which try to explain us the exact biology of the microglia and brain immunity. So this was our very significant observations, because that time we were working on a mouse model or aging model. What we could find, like in the aged mice brain, we were having a dot-like structure in the area, where in other like young or adult, they were having a different archaeological study. So those architect which were identified to be microglia, due to its activation, they are becoming round, because that is the property of the microglia. It undergoes different cytoskeletal rearrangement. So from a ramified to the activated round shape microglia. So this way we could identify it like it is present, and it is situation dependent of the brain that works there. So we try to look into that. Yes, if it is so and it's a versatile molecule, then how this PAX6 is known as a master regulator. So what it is doing? So we try to do chromatin immunoprecipitation, and by the PCR based identification, what we could find, like, yes, it is interacting to the promoters of the microglia, glia, and even like the genes which are associated with the oxidative response management. So from a normal maintenance to the oxidative stress dependent, it has a different activity, and it could give us a very promising observation which I would like to share, that it is binding to almost all promoters which are responsible for the functional anatomy of brain. So this is a data specifically doing chip, identifying those promoters and sequencing data. So these are the binding sites for BDNF, the catalase, GFAP, EBA1, PCNA. So you can have a look that it is affecting almost all genes. So it is again a situation dependent activity of this PAX6, that how it will be regulating even after the postnatal development. So what we could look into that, this is our recent findings. We were trying to explore it in a different model, which is well known as the T-cell lymphoma model, a Dalton lymphoma model. Many of you might be working on that. So we took that one as a model, like yes, if it is the immunologically challenges, what is happening in the brain of those mice which are having this lymphoma bearing capability. So we could find like it is again interacting with several such genes which are required for the immunological surveillance of the brain. And this way we could find like, yes, it is somehow this master regulator is working like a grid to regulate most of the gene activity. So what we have, another observation, we try to look into that, how it is, what is the mechanistic value of it. So we did a knockout, and what we could find, like it is associated or affecting most of the genes which are required for the maintenance of the neurons. So this is a Neuro2A cell line, and the knockout of almost all known siRNA of the PAX6, which we could customly prepared it. So we could find like it is affecting most of them. This is a summary slide of all those observations which I try to share with you, that it is working on the immunological surveillance, neural development which is there, and also neural degeneration, and also the energy homeostasis. We could publish and we could get several good or like encouraging comments related to that, and which gave us a little bit more idea to work on, and we try to explore some more facts. In that meantime, we are working on, like, since it is associated with the stress, oxidative stress management genes. So we try to explore that how and why it is working on it, because it has a binding site on catalase. So we try to give that cell line, to a different level of stress, like just treatment with the hydrogen peroxide. And then we try to look into that yes, it is working and it is stress dependent, the translocation of that paired box, that is PAX6, from nucleus to the cytoplasm, and this transit from nucleus to cytoplasm depends upon what is the status of the oxidative stress. So we could demonstrate that, and in that one we could find another important molecule, which is SPRC, which is working as a subtle protein binding with the PAX or interacting with the PAX, and depending upon the neurological microenvironment that is working in the brain. Other than that, like, since I tried to tell you this is as per theme of the conference, then since it is working with the oxidative stress management gene, so we could look into some of the facts, like, yes, if it is so, how those nutraceuticals can be useful for modulating the activity of specifically the microglia. So we have this is again a collaborative work. Again, Dr. Sasank worked on it. We published it, like microglia specific drug targeting using natural products for the regulation of redox imbalance in neurodegeneration. We try to look into the literature and our observations, and we could manage, like, yes, again, it is working at different level, and their interactome is very much important for almost all, whether in a disease condition and the microglial state. So this is again a summary slide which is already published, just to give you a introduction that yes, this is the mechanism, and at that point this PAX6 is interacting. Other than that, like, then we had a little bit more interest, and again Dr. Sasank worked on it. We could find, like, modeling of those receptors which are present on the microglia, and we try to look into some of these natural products specifically, and what we could find, like, those natural products, like, they are interacting with the microglial receptor, specifically CD40L, CD40L, and the CSF1R. So this way we could have some observations, and I would like to present a summary slide that yes, other, like, we are talking about curcumin. So from the morning we have several observations, and we saw that this curcumin is interacting specifically to the CD40L. Another than like, we have resveratrol and cannabinoids. Good thing about, like, maybe another we could get a patent on it, because many of you who are working on the curcumin, the major problem is, like, curcumin as such is insoluble. So we have collaborated with one of our colleague in chemistry, and he has developed a system what is basically, let me disclose it, it is referred as a QD, or quantum dots. So utilizing those quantum dots, we are able to solubilize this curcumin, and even 10 times more concentrated form of that curcumin can be used. We have very good preliminary data on it, like delivery system as an aqueous media. You do not need any of the media which is going to affect that cell, and we have submitted that for the patenting of these formulations. This way we could identify many of it. So after going through it, what we could find, like, as a summary slides, like, this PAX6 in between, it interacts with the P53, and also, like, this way it is working from proliferation, differentiation, and migration, and specifically during synaptic integration, SPRC is working at that, microglia has a major role of its activity, and this way it is working on both of these. I am not discussing here those signaling molecules which are like again MAPK dependent, and again like in both of them there is another very critical component, that is RAS gap. So by interacting with RAS gap, it is also working at the synaptogenesis and the protection of the synaptic plasticity. So that is another observations we have published, that it is expressed and it is interacting with most of those vital markers or proteins which are required for the maintenance of those cells, right from the olfactory lobe and hippocampus, amygdala, and cerebral cortex and cerebellum. This way, like if you recall that if there is no PAX6, or if the PAX6 is in less amount, it has different level of the impact, right from a mental retardation to the autism. So this way the level of PAX6 and its expression and interaction at different level of the brain, it is affecting almost all activities of a human being or any of the organism models. And this was like mainly I tried to explain you the work of Dr. Ratnakar, Dr. Sachin Shukla, and Dr. Sasank, who is one of here in the audience sitting somewhere. And thank you very much for your patience.
Just one question from my side. This PAX6 is expressed at higher level in neurons or in microglia, or it is the same level of expression?
We have no such quantification.
Okay.
What we have, that it is expressed in almost all cell types, including microglia. Certainly it is again compartmentalization is there. So even it is expressed in olfactory lobe, cortex, and cerebellum. That we have done. And also, like I tried to tell you in the different parts of the brain, it is expressing, including the amygdala. So microglia, it is expressing, that we have demonstrated, but which one is how much, we don't have this idea.
Because
but
you are connecting it with neurodegeneration, and that's why I asked.
Yeah, so it is again like maybe we are also discussing that something is threshold level.
Yeah.
That's why depending upon that threshold level, the way the microglia is acting as a maintenance or the activity as a protecting microglia, one aspect. And label, certainly, like if it is, we have some data, quantification data, that is in relation to other. So maybe like if 10 total I am taking, at least one of those express genes, say like EBA, is required for that 10 label. So probably it will be a differential expression pattern that we have not done, but it is expressed in microglia, and its activity is both at the protein-protein interaction level and also at the promoter level interaction.
Thank you very much, sir. Yeah. Any other question? If no other question, yeah, please.
Good afternoon, sir.
Good afternoon.
And thank you so much for providing this much information. There is a very little question from my side that, sir, during your observation and experimentation, what were the readouts which were confirming the autistic-like behavior in those animals, or the, you can say the behavior parameters, or how you confirm that this is autism?
See, like that autism is not our observation. That autism is well established related public data through which I try to give you like who has done it. So that work is based on several such patients who were having this autism and even their post-mortal brain. So they have identified that yes, it is the 3' UTR mutation of the PAX6. Almost all those patients we were having autism, they were having that part of the mutation. So that was the basis of the correlation between the PAX and autism.
Thank you so much.
Thank you.
So our next speaker is Professor Sharat Yasanti. He's a professor in the Department of Zoology at the Nagaland University, and his interest is neurobiology with special reference to Parkinson's. Is Sharat here?
Yep.
Oh, thank you. Sharat, stay with 20 minutes, please. With 20-minute time period.
Yep. It's slow. Yeah. Thank you for the time. At the outset, I take the opportunity to thank the organizers, in particular Professor Alok and Professor Anju for the opportunity to come over and present the little work what we have been doing.